evening, everyone. Thank you so much for being here. Tonight, we are going to do an all-in-one on a scientific reasoning in preparation for your TEAS exam. I know I worked with a few students that are getting ready to take it Christmas Eve, so I'm sure this will be very timely for many of you. As we go through this tonight, just know that as we have multiple choice questions for you to interact and participate with, if you are in Zoom, your answers are private, only I see them. If you're on YouTube or Facebook, those are public answers, but please do participate. That will make that much more enjoyable for everyone, and it will also help you stay engaged. So let's take a look before we dive in at just all the different things here at Archer that we have available to you. As you know, we have the free live webinars. This is the fourth in the science series. There's an anatomy, there is a chemistry, a biology, and then scientific reasoning. You can find out where all of those live webinars are coming up, doing that QR code. You can also re-watch old webinars using this QR code. And then free to you, those are all free to you. Then also free to you is our question bank. And our question bank has over 1,500 practice questions that really resemble the type of questions that you're going to see on the TEAS. And it also gives you rationale so that you can learn, if you miss one, why a different answer is the better answer. We also have on-demand videos. So the on-demand videos are something that you would purchase that takes you deeper into the content than we're able to do in these all-in-ones on the webinar. We have tutoring. As I mentioned, I have tutored several people in preparation for a TEAS that they're going to take on Christmas Eve. So that's available as well. And then we have bundles. We have a SurePass bundle, which is the question bank and the on-demand videos. And then we have the SurePass Pro, which includes tutoring. So these are all things you can access on the Archer website. And those are great packages. You get a little bit of a discount that way, and it helps you challenge yourself with the questions. You can track your performance, and you can see targeted videos for specific goals and objectives. So just a really exciting thing to offer for you here from Archer Review. Also, if you need anything related to TEAS, you can reach us on Instagram, TikTok, Facebook, and you can even email us at freenursing@archerreview.com. So we have many, many different ways that you can communicate with us. As far as tutoring is concerned, there are 3 of us based on your content needs. So Miss Tammy is our English and reading specialist, so you could scan that QR code if you would like Tammy to do some tutoring for you. I am Shelley Harshberger. I am our science expert when it comes to tutoring. You can scan that QR code for science tutoring. And then Mr. Brandon, he is our math guru, and you can scan that QR code for Mr. Brandon as well. And these QR codes, as well as the QR codes for all of our services, will be available at the end of the all-in-one webinar this evening. I also will have the slides that I share with you available at the end of the webinar as well. So let's get started. So our topics tonight that fit into the scientific reasoning category include scientific measurement and tools, evaluating scientific information, predicting relationships, and interpreting scientific research. So our first one aligns with S4.1. This is mastering a scientific measurement and the tools that are used within scientific measurement. So our learning objectives in this first category include understanding the units of measure and performing conversions within the metric system. I think that's something that is so critical here in the US, where we don't really use a lot of metric system, but in the medical field, you will. Our next learning objective in this particular segment is to identify the units and the numerical values in things such as diagrams, illustrations, graphs, or other forms of models. Being able to select the appropriate tool in order to measure length, mass, volume, and then finally looking at the appropriate scale based on the object being measured. So the metric system may seem a little less important in the US because we don't use it as often. But when you're thinking of the medical field, it's essential to ensure consistency, to ensure precision, because when we're talking medical, we're talking global. And we're one of very few countries that actually do not use the metric system. Most countries do use it wholly. And so it's very important that when we think medically, we think globally, and that we have a universal standard to ensure that consistency and to ensure that precision. And so that's very important for global communication in the medical field. So the metric system is also known as the SI, or Systems International. And it uses base units and prefixes to adjust the size or the measurement. And so this includes grams for mass, meter for length, second for time. And then volume is actually calculated by multiplying length times width times height, and then you get a number that is cubed. And that is the unit for volume is liter. So the units of measurement actually can be memorized by this silly little sentence: King Henry died by drinking chocolate milk. Now, what does that mean? It means that kilo stands for king, hecto Henry, deca died, base by, deci drinking, centi chocolate, milli milk. And remember that in the metric system, these are values of 10. So they decrease by values of 10 as you move from left to right. So kilo is 1,000. Hecto decreases by a unit of 10, so now it's 100. Decreasing by a unit of 10, it's now 10 for deca. Decreasing by that unit of 10 for the base unit is 1. Decreasing by a unit of 10 is 0.1 for deci. Decreasing by another unit of 10 is 0.01 centi. And then another decrease of 10, 0.001 for milli. Now notice that all we then change is the base unit. If we're measuring length, we're in meters. If we're measuring time, we're in seconds. If we're measuring mass, we're in grams. So we can convert then from meters to millimeters. We can convert different things based then on the system. So for example, if we have 5 meters and we want to convert that to millimeters, we want to move the decimal places 1, 2, 3. So 5 meters times 1,000 for the 3 decimal places makes 5,000 millimeters, okay? Or if you had 5,000 millimeters and you wanted to make it meters, you'd move that decimal place to the left and you'd come up to your 5 meters. Okay, so that's how you do that. So we're gonna practice it now with 6 kilograms to centigrade. So we wanna find kilo on the chart, that's right here. And then we want to count over till we land to centi. So we're going 1, 2, 3, 4, 5. And as you move, then you fill in those spaces with zeros. Notice we went from 6, 1, 2, 3, 4, 5 zeros, and that's where our decimal place fit. See that? So we started with a decimal place of 6, would have been just 6 point. And we got 5 zeros after it. See how we counted over? We went from 6 to 60 to 600 to 6,000 to 60,000 to 600,000. So 6 kilograms becomes 600,000 centigrams. All right, let's practice a potential real life scenario for you if you were in the nursing field. So a patient's chart says that you need to administer 0.25 grams of penicillin every 4 hours, but the pharmacy only makes 250 milligrams penicillin tablets. How many tablets should you give in each dose? So you've got 250 milligrams, but you need 0.25 grams. So this is what we're starting with. See how we do that? So 250 milligrams is actually 0.25 grams. See how we could have just kind of went backwards from that case. So that way you would just be administering 1 tablet. So then other things that you would need to be aware of is measuring distance such as end-to-end distance. So in that case, we would use a ruler or a meter stick to measure width, height, and length of an object. And we always want to start from that 0 point. And I think that that is something that is really important for us to remember when we're measuring, as we want to start right there. Okay, we want to start right here. And I think that that's an important thing. It's easy for us sometimes to want to just put the end of the meter stick up against something, but we have to be attentive to the fact that we want to start where the 0 is on our meter stick. So it's right there, okay? Most rulers or meter sticks are going to display each numbered mark as a centimeter, okay? So that's 1 centimeter, 2 centimeter, 3 centimeter. The smaller lines in between are going to represent tenths of centimeters or millimeters, okay? So if the measurement reaches the first mark after the 1, so let's say the measurement reaches right here, then that would be 1.1 centimeters or 11 millimeters. Now, if we want to measure mass, there are 2 tools that are common for measuring mass, and these are balances. One is the triple beam balance, and the other is the electronic balance, my bad. A triple beam balance, believe it or not, you're probably familiar with this, whether you know it or not, because a triple beam balance is basically what a doctor uses in the doctor's office when they weigh you. It's very, in a scientific realm, in a lab, it's not as common to use, but doctors do use it when they weigh you. Now, an electronic balance is something that you'll have more experience with in the field. And this is going to provide a digital readout, and that is more common, and it will do the measurements in either grams or kilograms. Now, as I said earlier, measuring volume is actually a mathematical formula, and it's measuring the length times the width times the height of the object. And then that will come up with centimeters or meters cubed. And that would be your unit then for measuring volume. Now, measuring the volume of a liquid is a little different. We generally use graduated cylinders for that, things like, or graduated containers like graduated cylinders, which is what's pictured. We can also use beakers, we can use measuring cups. And the way that you measure the volume of a liquid is at the meniscus. So when you look at liquid in these graduated cylinders, you'll see that there's actually a curve there. The liquid is not just perfectly straight across. And so you want to read it at the very bottom line. So we see that right here with our arrow. We want to measure it right here. Okay, so in this particular example, our units are going by one. So this would be 1, 2, 3, 4, 5. So if we get all the way up here, this is 20, 21. Then if we just look at it a little closer here, we're 21 and something, probably 21.6. And our unit there would be milliliter. But you always want to read it at the bottom of that meniscus. That's how you read the volume of liquid. Now, we need to be logical when we choose our unit. So, for example, kilograms are going to be the ideal unit for masses like my Great Dane. So this picture here is my Bella. She's laying at my feet right now. This is Bella. And she is about 118 pounds. And I should know that in kilograms. Shame on me. But she would be something that you would measure in kilograms, not grams, because it would be way too many zeros. So you try to be very logical about what unit you use based on the object. On the flip side, if I wanted to measure just a small amount of liquid, as is pictured here, I want to do that in milliliters because I'm just measuring like a drop of it. If I was measuring a tiny object, such as the ladybug over here in the corner, I would measure that in millimeters because it's small. Now, if I want to think about measuring something such as textbooks pictured here, then I would measure in centimeters. Meters are going to be something that we would use to measure like a building height, length of a car, things like that. And then kilometers are something that we would measure long distances, like between cities, or if you think about people who run a 5K, that K stands for kilometers. So that's a distance that they're running. And so that would be the unit that you would use for measurement from start to finish in their race. So we're wrapping up this particular topic and we've got some key takeaways. The metric system or the SI system is going to be used to measure mass in grams, length in meters, time in seconds, and volume in liters. From large to small in the metric system is kilo 1,000, hecto 100, deca 10, base 1, deci 0.1, centi 0.01, and milli 0.001. Now we have a mnemonic that can help you remember that. King Henry died by drinking chocolate milk aligns from large to small with the beginning letters of each of the categories in the metric system. Remember that rulers and meter sticks are going to be used to measure length, width, height. We're going to use volumetric flasks for liquid volume, like graduated cylinders and beakers. And then we'll either use a triple beam balance or an electronic balance for mass. And then we want to make sure that we are selecting the appropriate units based on object size. So millimeters for small items, kilograms for larger masses like my Great Dane, and kilometers for longer distances, such as running a 5K. All right, we've got some questions here. So our first question, and again, I encourage you to put your answers in the chat on Facebook or YouTube or our Zoom. Which of the following values is appropriate for the height of a refrigerator? Would it be 2.5 centimeters, 2.5 meters, 2.5 kilometers, or 2.5 millimeters? Now, I typically give you 45 seconds to answer these. So I'm going to go ahead and get my timer going now. Get those answers in the chat box. I'm getting some answers. Woohoo! Okay, many of you had this correct. If you chose B, you are correct. Well done. Now, centimeters would be too small. That would be something you would use to measure maybe the length of a book. Kilometers C would be something you would measure the distance between two cities, or like if you were preparing to run a 5K, that would be a half of a 5K. And then D, millimeters. That's something like measuring a little tiny bug if we had 2.5 millimeters. All right, let's do another. Which of the following is the meaning of the prefix milli? Is it A, 0.1, B, 0.01, C, 0.001, or 1,000? You've got 45 seconds. Put that up there to help you. King Henry died by drinking chocolate milk, for those that are wondering just quite which one would be the correct answer. Okay, if you chose C, which I think most of you did, well done. That is the correct answer. Remember that A would be deci, because remember B would be a unit of 1, 0.1 would be deci, 0.01 would be centi, 0.001 would be milli. Now the 1,000 starts with our kilogram, or our King Henry in our example here. Well done. I think most of you, if not all of you, had the correct answer. All right, let's do one more. Which of the following tools would be most appropriate for measuring the volume of a liquid? Is it A, an electronic balance, B, a graduated cylinder, C, a stopwatch, or D, a thermometer? I think everybody got this one correct. It is B, graduated cylinder. Well done. Electronic balance would be measuring mass, and stopwatch would be measuring time, and thermometer would be measuring temperature. So very, very good job. And I lied, I guess. We've got one more after this, and you might want to open to a calculator on your phone, or if you got one nearby on your computer. This one is going to be one where you might want to plug that in. All right, so a wooden block has a height of 6 centimeters, a width of 5 centimeters, and a length of 3 centimeters. What is the volume in cubic centimeters of the block? Is it 24 centimeters cubed, 32 centimeters cubed, 90 centimeters cubed, or 120 centimeters cubed? You got 45 seconds. And you guys are on fire tonight. I think everybody had the correct answer here as well. It is C, 90 centimeters. And remember, you would multiply 6 times 5 times 3. 6 times 5 is 30, and then 30 times 3 is 90. And that's how we get our correct answer. So very well done. You guys are on fire. All right, we're going to move into our next subject. This is using evidence and logic to evaluate scientific information. This aligns with S.4.2 in preparation for TEAS. So our learning objectives in this particular category is to be able to look at logical conclusions that we can draw from evidence that's going to be presented maybe in text, graphs, charts, some sort of visual. We want to be able to draw logical conclusions. We want to be able to recognize cause and effect in a scientific explanation. We want to apply evidence to support a scientific explanation. And we also want to be able to look at data and make really informed conclusions based on scientific evidence. So one of the primary features of science is that it relies on empirical evidence. That means that it's relying on information that's obtained either through scientific direct observation or it's obtained through experimentation. And that is important because it distinguishes it from a thought or an idea that's just based on theory or opinion. So science says, I'm not going to put all my eggs in one basket, so to speak, until I see the evidence, until I have a direct observation, or until I can experiment and come up with the same results over and over. So scientists are going to collect, they're going to scrutinize and analyze and draw conclusions based on large data sets. So they're not going to just assume that 5 little participants will support their conclusion. It's going to be a bigger group of participants or a bigger data set, whatever their focus is. And so then confidence in those conclusions is going to come from the fact that not only do they have a large data set, but that they could perform that experiment multiple times and still get the same results. And those are all important for us to understand so that we can rely on science versus an opinion or a thought or a theory that might be generated by someone very smart, but they haven't done this type of background work. And what science typically does is it really strives to answer or look at cause and effect relationships. It wants to see if this particular factor, the cause, directly influences or generates an effect. And this helps scientists explain phenomena, and it helps them make predictions about things that could occur based on the cause and effect relationship that's already been established. So an example of this, we're going to read through this together, is that scientists conduct a clinical trial testing a new medication for lowering blood pressure. One group is going to receive the medication, while another group receives a placebo. Now, if you are not familiar with a placebo, what a placebo is it is generally, in a medical perspective, like a sugar pill. It's not the medication, but the person receiving it doesn't know that. And a lot of times, neither does the researcher. And the thought process is there that if you've ever heard the phrase mind over matter, well, we can often kind of will ourselves to feel better because we want a new medication to work. And so they've got to offer this placebo to allot for that so that there's no bias in the study. So in this particular study, we're looking at one group's actually getting the new medication for lowering blood pressure, and the other one's going to get that fake pill. And it goes on to say, if the medicated group shows a significantly lower blood pressure, then the cause and effect relationship is clear. That the cause, taking the new medication, ends up with the effect of reducing blood pressure. Now, it's important to know that when researchers do something like this, they also have to control all other factors so that there isn't something else influencing the lowering of the blood pressure. So in this case, everybody that participated would have to have their diet, activity level, and existing treatments consistent. And that way, scientists could confidently attribute any blood pressure reduction to the medication and not that they suddenly started exercising or not that they started eating healthier. So they have to control and keep all those variables the same and only change one variable, that medication, to assess if it's effective or not. So the key components then of an experiment include things like independent variable, dependent variable, experimental group versus control group, and then the placebo. So let's look at these. So there's a relationship between the independent variable and the dependent variable. So the independent variable is what is being changed. So the independent variable is what is being changed. The dependent variable is what is measured in the experiment. So it's going to change in response to the independent variable. So it's what's being measured. So in our previous example, the independent variable was our blood pressure medication. The dependent variable was our blood pressure in our experimental participants. Now there's a relationship between experimental group and control group. The experimental group is the group that's receiving the treatment. So in our blood pressure example, the experimental group actually got the real medication. The control group is the group that does not receive the treatment. So in that case, that's the group that's getting the placebo. The control group is getting the placebo. And remember, I said the placebo, they generally just call it a sugar pill. It's an inactive substance that's just meant to compare the experimental group to the control group and help prevent bias. So let's look at an example and see if we can figure out independent, dependent variable, experimental group, control group, and then the role of the placebo. So let's take a look. Researchers are conducting a study to assess the effectiveness of a new supplement in improving memory recall. Participants are randomly divided into two groups. One group receives the supplement, and the other receives a placebo. Throughout the study, participants' diets, sleep schedules, and cognitive activities are controlled to avoid external influences. At the end of the study, participants are given a memory test to assess recall abilities. If the group taking the supplement performs significantly better on the memory test than the placebo group, researchers can conclude a cause and effect relationship. Cause, taking the dietary supplement; effect, improves memory recall. By controlling other factors and repeating the study with similar results, researchers aim to confirm the supplement's effect on memory enhancement. Now I'm going to set the timer and you can just kind of think about this and then we will go over the 5 questions: independent, dependent, experimental, control, and placebo. So you've got 45 seconds to think about that and then we'll reveal some answers. Okay, so let's take a look here. So the independent variable was the supplement. The dependent variable is going to be the performance, the memory recall performance of the participant. The experimental group is actually getting the supplement, the control group, the placebo. And then the role of the placebo in the experiment is to ensure that any effects are due to the supplement and not psychological. Like I said earlier, mind over matter, that's a psychological factor. And so we want it to be totally aligned with the supplement, not with some psychological response. So someone noted here that they attended a previous webinar and did not receive the slides. So the slides were a URL link at the end of the webinar. And that's what the link will be here as well. So hopefully that answers that question. All right, so let's take a look at analyzing experimental design. So we want to be able to analyze experimental design to ensure that the conclusions are reliable. So for example, in the memory recall experiment, a larger sample size, repeated trials would occur to increase our confidence in the supplement's effectiveness. If we only did one trial, it wouldn't be as effective. If we had a small number of participants, it wouldn't be as effective. But if we were able to have a large sample size and repeat it over and over and have the same results, that would give us confidence that the supplement really works. We also have to remember that all conditions except for that independent variable must remain constant. Okay, if we change anything else, we don't know. Is it the variable we changed or is it the supplement? So we got to control variables like diet, sleep schedules, everything should be the same across the group to ensure that observed effects are due to the supplement. So I'm still seeing some feedback here. The webinar ended and they weren't able to get it. So I'm sorry to hear that, but you'll certainly get tonight's. It'll be a URL when we're done. That's awesome. And if you want to send an email to the email at the end of this, I can see if I can get you those slides. So I don't have those slides right with me tonight, but if you want to send an email, I'm happy to try to get those to you. No problem at all there. Okay, so I've talked a little bit about bias. So let's talk about what bias is and how we want to make sure we avoid it. So bias refers to any type of influence that's going to affect the outcome of the experiment. And that could be based on the researcher's expectation or the participant's expectation. And that's why I said earlier that it's very common for even the researcher not to know which group got the placebo and which group got the actual medication. And that's what they refer to as a double-blind study. It means both the researcher and the participants do not know who's getting the real medication, who's getting the placebo. That way, bias isn't entered into it. That way, no hope of perceived benefits of the medication is going to influence how the reporting is conducted or how the data is gathered. So an example of that really is that the participants in the memory study might believe their memory is improving just because they're taking the supplement. Or the researchers might unconsciously interpret that the memory test results are more favorably because they know the supplement's being received. And so to avoid that, they'll just do blind studies. So as an informed reader and as someone whose future is going to be in the medical profession, it will be important for you to recognize bias in experimental design. So you want to look for a random assignments of participants so that we don't have skewed comparison. So that means that unless there's a particular age range that the medication is suited for, we need all age, we need all sexes, we need all races, all cultures, so that we can have comparison, we can have results that apply to everybody, not just one age range or not just one gender or one race or one culture. We want to make sure that the study is using some sort of blinding, and we're going to have more confidence in it if it's a double blind, because then even the researcher doesn't know who's getting that medication. And we really want to be aware of potential conflicts of interest. So if a medical company is funding the research, it does, put a little bit of a red flag there. That can influence the results. So you really want it to be a neutral party that is funding the research or a neutral party that's conducting the research. So those are things to be aware of when recognizing bias in experimentation. Another thing that's very, very important to understand, especially in today's realm with social media, is that we have to let science supersede opinion. Science has to be held at a higher level of standard than opinion because it's based on empirical evidence. It's based on direct observation. It's based on repeated experiments. And so that is proof that the science at that time is greater than just someone's personal beliefs. Scientific claims are reproducible, they're testable, they're objective, they try to minimize and avoid bias as much as possible. And then, even after they go through all of that, the data and their whole process is just scrutinized by experts, peers, before it is accepted and before it is published. And so it's such a rigorous vetting process that we have to understand that science is held at a higher regard than opinion because of that. And it's also important to understand that science is evolving. We've got new evidence, we've got new ways of looking at science. And so, if something that we once believed is now slightly different or tweaked or whatever, it's not that science was wrong per se, it's just that science learned more. You know, case in point, we see all the time with like cold case files when they couldn't assess DNA, and now they can assess DNA and catch the bad guy. And it wasn't that they were wrong back in the day, but they just didn't have those tools of DNA to assess at the time. So those are some things to think about. So I see a question. I'm not sure I fully understand. It says how science controls bias by controlled variables. So we control bias by trying to remove any personal beliefs or opinions. Controlled variables is something that we try to keep steady so that the only variable that changes is the one we're measuring. I hope that answers that question. All right, so some key takeaways for this segment. Remember that empirical evidence is data that we're getting from experiments or direct observations. Data analysis, these are conclusions that are drawn from large data sets and from reproducible results. Remember that cause and effect is really what scientists are striving to determine. So they want to understand the relationships to help explain phenomena and to help predict outcomes of similar experiences in nature. Some key terms that we have, independent variable is what is being changed or manipulated. They use that word manipulated. It just means that that is what's being changed. The dependent variable is what is being measured in response to the independent variable. So it's the measured outcome. The experimental group is the one that's getting the treatment. The control group is what's getting the placebo. I remember the placebo is generally just a sugar pill. It's some sort of inactive substance given to that group, and no one knows, hopefully, in a double-blind study or in just a blind study, the participants don't know. Remember that bias is any influence on an experiment due to like psychological factors. It could be expectations of the participants, it could be expectations of the researchers, and we want to try to avoid that. And as a future medical professional, you want to be able to really recognize bias. You want to look for the fact that there are random participants, that there's blinding in it, and you want to be attentive to any potential conflicts. All right, let's do some practice. So this first slide just gives us information, and then we will ask questions from that point forward. And the questions do have the data with it to help us best answer it. So let's take a look at our practice problem. A researcher conducted an experiment to test the effectiveness of a new blood pressure medication. 4 patients with elevated blood pressure were selected for the study. Each patient was given a dose of medication at 8 a.m., and their blood pressure was measured immediately. The patient's blood pressure was measured again at 11 a.m. to assess the effect of the medication. Patient 1, however, received a placebo instead of the medication. The other patients, 2, 3, and 4, received the same dosage of the new blood pressure medication. The results of the experiment are shown in the data table. Use this data to analyze the effectiveness of the blood pressure medication over a 3-hour period. Now, this data will be on our next slide as we look at the questions. Which of the following conclusions can be drawn from the data? A, the blood pressure medication reduces patients' blood pressure after 3 hours. B, the blood pressure medication does not affect blood pressure levels. C, in large doses, the medication is effective. Or D, the correct time to take blood pressure medication is 9 a.m. You've got 45 seconds. Okay, most of you were very successful telling me that the correct answer was A. So what we see here, let's take a look at this to help break down some of the other responses. So number one is the placebo. So this person did not get any medication, and you can see that at 8 a.m. and at 11 a.m. their blood pressure didn't change. Now 2, 3, and 4 all received the medication. This was their starting blood pressure. This was their ending blood pressure. So they were on the medication for 3 hours when we look at their results at 11 a.m. So person number 2 started with a blood pressure at 155 and it went down to 140. Patient number 3 started at 162 and it went down to 130. Patient number 4 started at 150 and it went down to 145. So each of these patients' blood pressure did reduce after the 3 hours. B says that there was no effect. Well, we did see an effect. C, in large doses, the medication is effective. They didn't say anything about doses. We are to infer that they gave everybody the same dose. And then D, that the correct time to take blood pressure medication is 9 a.m. Nothing was done at 9 a.m. So that's just kind of there to throw you off. All right, so in this scenario, it gave us the same blood pressure background with a new question. So since we already read it together, I'm going to leave it up, but let's take a look at our question. Which of the following describes what was done to try to avoid bias in this experiment? A, all patients were given the medication when blood pressure levels were highest. B, all patients had similar starting blood pressure levels. C, a placebo was given to the patient who did not receive the medication. Or D, the experimenter followed the same directions each time when administering the medication. Go ahead and work on that and put your answers in. You got 45 seconds. Okay, and I think everybody was also correct on this one that the placebo was given. And that is what's helping reduce bias. Now, I did have a question or an observation, and I do want to go back to it because I think it's a valid observation. Someone noted that the proportion of blood pressure decrease was not the same in every individual. Like this one, the blood pressure only went down 15 points. This one, it went down, 28? Here it only went down 5. And that's the human factor. So the blood, just like the blood pressure didn't start at the same level, it's not going to necessarily be as effective for every single person. There are going to be lots of different factors. Genetics plays a role. Some people can have the healthiest lifestyle. Some people can have the healthiest diet. And genetically, they're just predisposed to have a high blood pressure. So that kind of factor could factor in. Poor diet could factor in. Many other factors could impact why the proportions aren't the same. But the fact that everybody's blood pressure did decrease leads us to believe in this very small sample size that it reduced blood pressure after 3 hours. So good observation there. All right, so we still have the same scenario, but we have a different question. The experimenter instructed patients to maintain similar activity levels during the 3 hours of testing. Which of the following is the name for this type of experimental factor? Is it A, data, B, independent variable, C, dependent variable, or D, controlled variable? You've got 45 seconds. Okay, so for this one, hopefully, I think everybody chose controlled variable. So these are the factors that they're keeping all the same. Okay, I see I do have a question for the previous one. The answer was placebo. Let's take a look here. So they're wanting to know why it wasn't D, and that's a very good question. So this previous one, it says, you know, what they were doing to avoid bias. And D says the experimenter followed the same directions each time when administering the medication. And they did do that. That is true. But that would be more like controlling the variables. Our next question, not about avoiding bias. Bias is psychological impact. And so they're trying to avoid expectations or psychological impact of it. And where D is they're keeping everything the same. So I hope that helps clear that one up. All right, I think this is our last one using the same scenario. Let's take a look. So same scenario, different question. Which of the following experimental practices would strengthen the data supporting the conclusion? Is it A, testing multiple dosages of the medication? B, testing the medication at different times of the day? C, testing more patients with similar blood pressure issues to see if the results are reproducible? Or D, removing the placebo group from further testing to prevent bias? You've got 45 seconds. And I think many of you had this. Well done. The correct answer is C. And that idea is about being reproducible. So the problem here is multiple dosages. They didn't talk to us about dosages at all. So basically, that is something they could do later on after they've established that the medication is effective. Then they could play with dosages. But for right now, they're just trying to see if it's effective or not. So in that case, the key is it reproducible? Testing the medication at different times of the day. Again, we still go back to, we just want to know, is it effective? Can we reproduce effective results? And then we can assess if different times of the day matter, if different dosages matter, that sort of thing. And then D, we definitely do not want to remove the placebo. The placebo then helps prevent psychological impact. And so we don't want to remove that. Okay, we're moving right along here. We're moving into our third of 4 categories. This is being able to predict relationships in events, objects, and processes. So our learning objectives here is that we want to evaluate the size or magnitude of an object or process. We want to identify the different causal relationships in certain events. We want to look at the logical sequence of events in a given situation. And then we want to assess the impact of events compared to others. So let's take a look. So magnitude of size. So what this means is that we need to understand the scale and the units of measurement in order to understand what tools and what methods should be used for patient care. So for example, you wouldn't use the tranquilizer dosage on a cat that you would use on an elephant. So that's understanding magnitude of size. We're going to use a very different amount of tranquilizer on a cat, a much smaller organism than an elephant. Or we wouldn't use a scalpel to remove a femur. That's way too small. But I'm sorry, we would use a scalpel to remove a femur. No, I'm sorry. I was trying to read that and it was not making any sense to me. My bad. So yeah, using a scalpel to remove a femur is definitely too small. But yeah, the scalpel is too big to try to cut an atom in half, something we can't see with a naked eye. And so we have to make sure we're using the right tools for the job. And that's the take home message. And sorry about that. That was kind of a mess. All right, moving right along. So this is where we have to think about size of things to really get an idea how they would be measured, what units we would measure. So an atom, something we cannot really see with the naked eye, that's going to be measured in a nanometer, which is a billionth of a meter. Imagine that. A molecule such as a water molecule pictured here could be measured in a nanometer or more than likely in a micrometer. Okay, and a micrometer is a millionth of a meter. Now, an organelle, something that is inside of our cells, could be measured in a micrometer as well. And even some cells could be measured in micrometer as well. It's not until we get to tissue that we start looking at measuring it in millimeters or centimeters. And a millimeter is 1,000th of a meter, a centimeter is 100th of a meter. Okay, so that would be, like the tissue that forms an organ, or the tissue that's part of our skin, that sort of thing. If we took a biopsy, let's say we were worried about skin cancer, we would maybe measure our biopsy in millimeter or centimeter. Decimeter then, which is 1/10 of a meter, is something that we might start using when we measure the heart. The heart could be measured in decimeter. It might still be measured in centimeter, but that's an organ that we could kind of measure in either unit. Now, meter itself is going to be used when we're measuring an organ system or the organism itself. Okay. And this is another one where if we want to remember, if we want to go through these orders of measurements from nanometer, micrometer, millimeter, centimeter, decimeter, meter, we can use another silly sentence. Now for nanometer, many for micrometer, millipedes for millimeter, crawl for centimeter, down for decimeter, mountains for meter. Now, many millipedes crawl down mountains. So that would be a silly sentence to remember these really small measurement units that lead up to the meter, such as measuring a human. All right, so this is a fun one. It's not one you can put in the chat, but it's one you can think about as you go through. So it wants us to match up the structure to the type of unit we would use. So red blood cell, human height, human heart width, atom, human torso length. So you got 45 minutes and then we'll reveal some answers and you can get a sense for how you did. Okay, so let's see how we did here. Red blood cells, that would be measured in micrometers. Human height in meters. Human heart in centimeters. Atom, nanometers. Human torso length, decimeter. So I've got a question here asking, reproducible here means same result. Yes, so if we did an experiment, we reproduced that experiment and we got the same result, that would give us confidence that the experiment, that our findings in our experiment were accurate. How can you remember that? Ooh, that's a good one. To reproduce something means that you make another one or you do it again, and in this case it means doing it again, and because you hope that it is going to give you the same results, that's where we're getting reproducible. That's the best I got for you. That's not a question I've mulled over at any length. I apologize. All right, so now magnitude of process. I think that even the title of this one can be like, so let's take a look at our example here, and this will make sense. It says it's an example of comparing the magnitude of processes is the comparison between aerobic respiration and anaerobic respiration. So let's talk about what they're talking about here, because this might be foreign to some of you. So if you remember from high school biology, you might have looked at something called cellular respiration. And collectively, that's what we have on the screen here. And cellular respiration is where your body takes food and transforms it into the fuel that you need to function. And the fuel is ATP. So anytime you see ATP, think body's gasoline. It is the fuel. And so the magnitude of process here, what they want us to understand is in the presence of oxygen, that's what aerobic means, we get a lot more fuel. We get 36 units of ATP versus in anaerobic respiration, which means without oxygen, we only get 2. So the magnitude of process is that there's a huge difference between the energy that we can generate from our food if we have oxygen present versus the energy we can generate from our food if we don't. And that's what they mean when they talk about magnitude of process. We obviously want to have oxygen available so that we can get the most bang for our buck. We can get the most fuel for that food item. That's what they're getting at. All right, so sequence of events and causal relationships. So as we said earlier, scientists are really looking to answer, cause and effect. If I do A, will B happen? And so they'll infer causal relationships by gathering data through, as we said earlier, experiments or observations. They're going to thoroughly record what they see. So they're going to record patterns. They're going to record trends. They're going to record every meticulous outcome they possibly can record. They're going to examine the sequence of events to ensure that the potential cause actually comes before the effect. They're going to manipulate just one variable at a time, the independent variable, while keeping all the other conditions constant. Because if we change multiple things, we don't know what helped us. Here's a prime example of that. Let's say you wake up tomorrow morning and you have a horrible headache. And you think back and go, oh, wow, I didn't drink any water yesterday. I'm going to take some Tylenol and I'm going to take my big Contigo with me and I'm going to fill it with water and I'm going to have lots of water today. And at the end of the day, you feel great. Was it the Tylenol or was it the extra consumption of water? You don't know because you changed 2 variables. So when scientists try to determine something, They want to only change one variable. So in my water example, you could wake up and say, oh yeah, I don't drink enough water. I've got a headache. I bet it's because I'm dehydrated. And you could drink a bunch of water that day, skip the Tylenol. And at the end of the day, if you're feeling pretty darn good, you can say, oh yeah, water helps and helps get rid of my dehydration headache. Because then you only change that one variable, that water. Okay. And then of course, conducting multiple trials. That's what our question was earlier about being reproducible. We need to do the experiment over and over and over to see if the same cause produces the same effect. That's that reproducible idea. Here are some examples of cause and effect. So poor diet and obesity. There is scientific evidence to support that consuming a diet high in processed foods and sugar, which is our cause, leads to weight gain, our effect. Lack of exercise and heart disease. A very mellow, sedentary lifestyle cause contributes to a higher risk of heart disease, effect. Exposure to UV radiation, prolonged exposure from the sun is a cause that can lead to skin cancer, effect. Smoking and lung cancer. Smoking cigarettes can lead to an increased risk of lung cancer. And I thought this data was really interesting. This is authentic data that shows how as cigarette smoking increased, these are the quantity of people smoking from the early 1900s well up into the 60s and 70s here, the prevalence of lung cancer also increased. And then as we became more intelligent about, oh, this is causing lung cancer, when people tried to quit smoking, then we see that lung cancer decreased as well. So we see here a visual of cause and effect between smoking and lung cancer. All right, something else that's important for us to understand when we think about cause and effect are something known as feedback loops. And you may be familiar with feedback loops when you've heard the word homeostasis. So homeostasis is something that a negative feedback loop aims to achieve. It's keeping a balance in our body. But feedback loops are really sequences of events that we can observe. So in this case, in a positive feedback loop, some sort of initial change causes a response that amplifies that change. In a negative feedback loop, an initial change causes a response that counteracts that change in order to maintain homeostasis, in order to bring us back to a balanced point within our body. So here we see examples of those. So a positive feedback loop is always going to amplify a change. So our example, our initial stimulus in this case is childbirth. The baby is pressing on the cervix. Our variable is that it's causing tension in the cervix. Our receptor that's detecting a change, our stretch receptors are stimulated. That sends a signal to a control center that evaluates that change. In this case, the control center releases oxytocin. Oxytocin then causes an adjustment, which in this case, it triggers uterine contractions. And that's causing the cervix to stretch more in response to the contractions. And that's our positive feedback. So it's amplifying that variable. So the cervix stretches, we've got more cervical tension, and we go through this cycle until the baby's born. And that is a positive feedback. We're amplifying that process. This, in that way, is not a basis for homeostasis until it's done, until it's over. Then the body would revert back to homeostasis. But positive feedback is not a homeostatic event. Positive feedback is where we're enhancing till we get to a set point. Now, our next example is all about homeostasis. So in this case, we've got a body temperature. So that's our initial stimulus. So we've got a cold environment. So then our variable is that our body temperature decreases. Then our receptor that's detecting the change, our thermoreceptors and our skin that say, ooh, we're cold. That sends a signal to a control center that compares it to a set point. In this case, the control center is going, ooh, we're not 98.6 degrees. What's going on here? And so the control center sends out, how can we get us back to good? Well, we can shiver and our blood vessels can constrict to try to get our body temperature back to normal. So in this case, the effectors are adjusting in the opposite direction of the stimulus because the stimulus was cold and this is trying to get us back warm again. And so in this case, the body temperature increases back to 98.6 degrees and we're back to a homeostatic event. Okay. So negative feedback associated with homeostasis, positive feedback, not so much. All right, we're wrapping up this third category here. Let's look at our takeaways. Biology from simple to complex. So these were those really small units. So we go from atoms, which make up molecules, which make up organelles, which are inside our cells. Like cells work together to form tissues. Similar tissues work together to form the organs. Similar organs work together to form our organ systems. And then all of our organ systems work together to form the organism. In that case, then, for the units for the simple to complex structures, we can use that mnemonic. Now mini millipedes crawl down mountains. And that stands for nanometer, micrometer, millimeter, centimeter, decimeter, and meter. Understanding measurement scales is important for patient care. As we said earlier, we want to administer the right amount of cation for the size of the organism. We want to make sure we're using the right tool, that sort of thing. And then scientists use sequence of events to ensure that the cause actually comes before the effect in a causal relationship. That's important. And that positive and negative feedback loops demonstrate the causal relationship through those sequences of events that we just went through. So if it's going to amplify the event, that's a positive feedback. If it's going to counteract the event, that's a negative feedback. All right, let's see what we know so far. We've got some questions here. Which of the following units is most appropriate for measuring the length of a human hand? A, centimeters, B, meters, C, millimeters, D, kilometers. You've got 45 seconds. Okay, I think most of you guys had that. Well done. It is centimeters. Remember, meters would be something you would measure like the human height in. Millimeters would be something that is much, much smaller, and kilometers would be measuring a distance like running a 5K. Okay, so centimeters. And I got some people, looks like on YouTube and Facebook. Thanks for joining and putting some answers out there. I appreciate that very much. Okay, let's see another. Which of the following units is most appropriate for measuring the mass of a human heart? A, grams, B, meters, C, kilometers, or D, millimeters? You've got 45 seconds. So the key word in this one is mass. Okay, so not sure why it made a big long line down there. The key word there is mass. And so because the keyword is mass, our correct answer is A, grams. Okay. Meters is measuring length and would be way too long anyways. Kilograms, remember I said that's like measuring the weight of us or my 120-pound Great Dane. And millimeters would be pretty small, and that's measuring length anyways, not mass. All right, which of the following illustrates a positive feedback loop rather than a negative feedback loop? A, insulin lowers blood glucose levels after eating. B, sweating cools the body down when overheated. C, heart rate decreases to reduce high blood pressure. Or D, oxytocin increases contractions during childbirth. We've got 45 seconds. Okay, I think everyone got that one right. Well, very well done. Yes. D is the only example that is enhancing to a point. So it's increasing those contractions. That is a positive feedback loop. Okay. Now I did see that I have a question asking about meter and centimeter for medium objects, and I think it kind of refers to our previous question here. So centimeter would have been an okay measurement for the human heart if we were asking to measure the length or the width of it. But because it's asking for the mass is essentially a measurement like weight. It's measuring the weight of the human heart. And so we wouldn't be using any form of meter in that context. So hopefully that helps. All right, next question. Which of the following is an example of a negative feedback mechanism? A, platelets releasing chemicals to attract more platelets during bleeding. B, increased heart rate to pump more blood during exercise. C, sweating to lower body temperature when it rises. Or D, release of oxytocin during childbirth to increase contractions. You've got 45 seconds. So the key here is that it is a negative feedback mechanism. So a negative feedback is trying to get us back to homeostasis or try to get us back to a balanced environment. And so in that case, C, sweating to lower body temperature when it rises, that's trying to get our body temperature back to its normal range of 98.6 degrees. All the others are enhancements. Platelets are attracting more platelets during bleeding, that's an enhancement. Increasing heart rate to pump more blood, that's an enhancement. Releasing oxytocin to increase contractions, that's an enhancement. Those are all examples of a positive feedback mechanism. All right, we are on our last quarter for the night, the final one. And this is application and interpretation of a scientific investigation. So in this case, we want to be able to form a hypothesis that relates to a given investigation. We want to be able to recognize a basic experimental design to test a hypothesis. We want to be able to determine dependent variables, independent variables, experimental controls. A lot of those things we've already talked about, but we want to apply them now. And then we want to evaluate whether the results of an experiment support a hypothesis or go against the hypothesis. And when they say go against a hypothesis, they say refute the hypothesis. And it could be going against a hypothesis, a prediction, or a conclusion. So this is really, really an interesting study. So back in 2016, NASA conducted a study and determined that only 28% of Americans could accurately assess how credible scientific information was. So what that's saying is that all this content that we read on social media, or that we read on news, or we read online, or we see on TV, really only 28% of the American public can truly make sense of that. And so that's why they add scientific reasoning to your TEAS exam, because as a future medical professional, it's going to be your role to help patients navigate and understand medical information, especially as a nurse. A doctor is going to leave that to you. A doctor is going to say, I've got Sally here, and Sally's going to talk you all through that, and he or she is going to move on their merry way. And so it's going to be really important for you to have a solid grasp of understanding how scientific experiments are conducted and how they draw conclusions that can be trusted and that you can have confidence in. And so that's why this is such a critical component of your TEAS exam. So we know that most scientific research begins by a researcher observing something, observing a phenomenon that they really don't know why it occurs. And they form a hypothesis then, which is really their best informed guess about a causal relationship. They think that, A causes B. And then they'll start going, okay, well, how could I do an experiment to determine if A causes B? And so the research then is going to determine if their hypothesis is correct. And then if it is correct, it's going to be supported by the data collected, or it's going to be refuted. The data is going to say, no, your best guess was wrong. And then if the data supports it, there's still going to be repeated trials. That's that reproducibility we talked about earlier, and a scrutinizing process through peers to provide validation, to ensure that the findings are accurate. They're going to challenge them to really scrutinize so that we can have confidence that the outcome is authentic. So the variables, we talked about some of these before, but I do want to add a couple things in there, so it is important. So remember, we said that we've got our factors in an experiment that are manipulated, held constant, or considered in an experiment. Those are all considered variables. And we said before that the independent variables, what you're changing, and then the dependent variables, which you're measuring based on that independent variable. But what I wanted to draw to your attention, and I think this is really important, is that the independent variable, if you were going to look at this on a graph, the independent variable is always going to be on the x-axis and the dependent variable on the y. Okay? And then we've got our constants, or what we call our controlled variables. Those are all the things that we're keeping that stays the same. Okay? Controlled variables, constants are staying the same. So like in our blood pressure example we had earlier, we wanted them to keep their same diet, their same activity level, their same sleep schedule. We didn't want any of that to change. I have a question here, which is, this is a good time to throw it in there. They asked about the difference between experiment and controlled variables. So controlled variables are the things that we're keeping all the same. Okay. The experimental variables are the things we're talking about here, such as the independent variable, the thing we're changing, the dependent variable, the thing we're measuring. Okay. And we're going to go into an example, and that may help clarify that as well. All right. So this is an authentic experiment that I conducted. And so let me just share. I'm a scientist by heart, and this was one way that I can share how it really happens, even in a home environment. So these pictures here are pictures of Cattleya orchids. I have grown orchids for many, many years. Despite the fact that I've grown them for many, many years, I don't fertilize them nearly often enough. And so I started noticing, my observation was that I wasn't getting flowers as often. So Cattleyas typically only flower once a year, but you can get many, many flowers at that time that they're blooming. And I wasn't really getting nearly as many flowers. So my hypothesis was, if I fertilize, then flower production will increase. So that was going to be the basis of my experiment. So my scientific hypothesis, just to make it more formal, I stated the addition of fertilizer will increase the number of flowers in orchid plants. So there's my hypothesis. And ultimately, my experiment is either going to support it or refute it. It's going to go and say, yes, my flower production did increase, or no, my flower production did not increase. So my experimental setup, I had an experimental group. I had 10 Cattleya orchids that I gave fertilizer to. Then I had my control group, 10 Cattleya orchids that did not receive fertilizer. Now, granted, I did not use a placebo here, but orchids don't have psychological effects, so I didn't have to worry about that. And for me, it was going to be black and white. Either there were flowers growing or there weren't. So it wasn't like I could have bias in that. My independent variable then was my fertilizer. My dependent variable, remember, that's what I measure. That's the number of flowers that were produced. And then my constants, that's all the stuff I wanted to keep the same. So they were all Cattleya orchids. They all got the exact amount of water. They all got the exact amount of sunlight. Their temperature was exactly the same. And what I grew them in was exactly the same. Fun fact about orchids, orchids don't, most of them grow in the ground. Most of them grow attached to trees. That's why you see them growing in a tree bark kind of base. If you go to Myers or Menards or Lowe's and you see orchids, you'll see that they're really kind of growing in tree bark. So that's why that doesn't say soil, it says growing medium, because they all had to be in their tree bark type growing medium. So then my data that I collected. So I collected data on the number of flowers produced by each group over 8 applications of fertilizer. Now, orchids, even when you do fertilize them, you can't fertilize them very often. It will burn them. So this was, I believe, like fertilizer once a month. So it was kind of over an 8-month period, right around that approximate time frame. And then I had to measure flower detection based on kind of where it was in its growing schedule. So I considered it a quarter of a flower if I detected it inside this little sheath. So when an orchid flower first grows, it grows in what appears to be a leaf that is folded over, and that's what's called a sheath. So if I could see that there was a bud in there, I considered that a quarter of a flower. If the buds, as you can see in this image, are peeking out of that sheath, I considered it a half of a flower. If I could start seeing what color they were going to be, if the petals were starting to be exposed up at the top, I considered that 3/4 of a flower. And then one was a fully opened flower. So over my 8 applications, we can see that this is the group with fertilizer. This is the group without. And we can see that really consistently there was more evidence of flowers right across the board. And the longer they were getting the fertilizer, the more flowers we could see. See right here, I had 4 total flowers detected where there was only like 1 3/4 there. So they just steadily increased as the application of the fertilizer occurred. So again, giving you a visual, because we often have to interpret graphs when we're making sense of data. So we can see that we've got our fertilizer application, that's our independent variable on our x-axis, and our dependent variable, the number of flowers, on our y-axis. Our independent variable is that fertilizer. Our dependent variable is the flower presence, okay? And we can see that the experimental flowers, there were certainly more flowers in the treatment group than the control group, the group that did not get the fertilizer. So our conclusion here was based on the data, the experimental group, the orchids with fertilizer, produced more flowers than the control group, the orchids without fertilizer. So that supported my hypothesis that adding fertilizer did increase the number of flowers in Cattleya orchids. However, I would say that this would not hold up under peer review or scientific scrutiny. I would need many more trials that would have the same results. That's that reproducibility that we talked about earlier. So we would have to have other people test out Cattleya orchids in the same fashion and get the same results. And larger sample size. I only had 20. Maybe they would have 200. I just used this as something I actually truly did, and it was easily understandable, relatable illustration of a scientific experiment. All right, so we got some takeaways. So many Americans struggle to assess scientific credibility. So that makes it really, really important for you, future nurses who are going to be on the front lines with these people, to have a strong understanding of scientific experimentation to help them interpret health information. Scientific research typically starts with an observation, an hypothesis is formed, and then it's tested through formal investigation. Key components of an experiment: independent variables, what's changed; dependent variables, what's measured; controlled variables is everything else that's kept constant. When reporting data, the independent variable goes on the x-axis, the dependent variable goes on the y-axis, and then many trials, peer review is necessary to confirm and find confidence in experimental results. Okay, we got some questions to wrap it up. A scientist predicts that plants grown in sunlight will grow taller than plants grown in darkness. This prediction is known as A, hypothesis; B, scientific assumption; C, controlled variable; D, constant. You've got 45 seconds. I think everybody got this one correct. A hypothesis. Okay, that prediction of the hypothesis. Scientific assumption is just there to throw you off. And controlled variable and constant are essentially the same thing. That's all the stuff that you're keeping normal, keeping the same, so that you don't have anything confounding your results. All right, when plotting research findings on a graph, which variable goes on the x-axis? A, the dependent variable; B, the controlled variable; C, the independent variable; or D, the constant variable. You've got 45 seconds. All right, I think most of you got this. So the x-axis is our independent variable. Okay, so this is our x-axis. This one is our y-axis. Okay, and the others don't go on our graph. Okay, now I did have a question. Someone asked if a scientific assumption and a hypothesis were the same thing. Scientific assumption was just a response thrown in there to throw you off. That is not a scientific terminology, really. That is just general language, but that's not necessarily a term or something that is used in scientific terminology. Hope that helps. All right, here's another. Which of the following is true about the dependent variable? A, it remains constant during the experiment. B, it is the factor measured during the experiment. C, it is tightly controlled by the experimenter. Or D, all of the above are true. You've got 45 seconds. Okay, hopefully you chose B. It's the factor measured. So the dependent variable is what's measured during the experiment. Okay. Constants, those are the things that we keep all the same, so we know what if the independent variable is actually impacting anything. Tightly controlled by the experimenter, that would be constants. And then of course D is ruled out. All right, I think this is our last one. In the orchid experiment, what variable was changed? A, the type of plant being used. B, the environmental conditions. C, application of a fertilizer. Or D, production of flowers. You've got 45 seconds. Okay, I think most of you got this one correct as well. It's the application of fertilizer. Okay, so that was our independent variable. That was the variable that was changed. Our dependent variable, if you chose D, was the production of flowers. Okay, so that was what was being measured. But what we changed, the cause of the production of flowers, was the application of fertilizer. Okay, so this was our independent variable. I'm writing this with my mouse. That's why it's not very pretty. And this would have been our dependent variable right here. Okay. All right. So just to wrap things up, I promised you'd see this slide again. If you want to look for our schedule of free live webinars and just get more information on our free webinars, that is the top QR code. If you want to rewatch old webinars, that is our second QR code. Our question bank, it's a free trial. You get over 1,500 questions with rationale as to why correct answers are what they are and why incorrect answers are what they are. If you want in-depth support, you can get on-demand videos, and these are very specific timestamps. So if you only have one topic, let's say you need to learn about mitosis, that's driving you crazy, you just can't figure it out. You would have a timestamp in that video that leads you right to mitosis. So it's pretty darn cool. As I said earlier, you do have an option of tutoring. I tutor science, and we have an English and reading tutor, and we also have a math tutor. That's our next QR code here. And then we have two different packages that are available to give you a little bit of a discounted bundle. We have a package that puts the QBank and the on-demand videos together, and we have a package that does QBank, on-demand videos, and tutoring. So all those QR codes are there to help you gain access to those different services. We're here to support all of your TEAS needs. This is just a visual of what you would see on the website that really dives into the two different discounted bundles. So the Pass bundle is the one where you're getting the question bank and the on-demand videos, whereas the Pass Pro, you also get that addition of a tutoring session. As I said earlier, you can reach us on Facebook, Instagram, TikTok, and you can also email. So that person who noted that they didn't get the slides URL from the November 19th session, email this email. Tell them that Shelly Harshberger was your host. I believe that was our biology session, and that you participated but just didn't catch that URL. And they would be happy, I'm sure, to get those slides to you, or get that to me, and I'll be able to get the slides to you that way. As I said, there are your tutors. You can scan here for any of us if you would like to book a session. And then finally, I want to get you tonight's slides. And while I'm getting you tonight's slides, I ask that you give us some feedback. We are here to serve you. We are here to help you with your TEAS needs. So feedback on tonight's session would be greatly appreciated so that I can give you the best all-in-one webinars going forward. So take a moment, please, and fill that out while I go ahead and grab those slides for you. Those slides will be available in the chat in both Zoom and Facebook. Oh, and YouTube as well. So I guess it would be more important for me to say all three instead of both. So get that survey going. I'm going to grab those slides. I'll be back with you shortly. All right, folks, the slides are out on the chat in Zoom, in YouTube, and in Facebook. So you are free to grab those. And I wish everyone happy holidays. And the next webinar that I'll have coming at you from me will be biology, and that will be in January. So I will see you in the new year. I'll leave this up just for a little bit. If you're just finishing up the survey, you can still access those slides. So I'll leave it up for a few more minutes. Have a lovely evening.